Okay, well, we're collecting people down. Let's start. Thank you everyone for joining the ISBS webinar for November 2025. This time we have invited Dr. Carl-Fredrik Burman to our session to talk about philosophical thinkings about Bayesian analysis. Before we introduce him, I would like to say that he would encourage a very interactive session today. So please put your thoughts and comments in the chat box, and we're going to pause from time to time to encourage comments and interact with the audience. So Dr. Carl-Fredrik Burman is Senior Director of AstraZeneca, Associate Professor of Chalmers University of Technology, and an affiliated researcher at Karolinska University. With more than 30 years of experience in the pharmaceutical industry, his focus is study design, methodologies, and multiple testing. He currently serves in the AstraZeneca methodology group led by David Wright. He recently completed his term as president of Swedish Society of Medical Statistics in the industry as the industry lead for the analysis work package in REALIZE-D and EU Rare Disease Research Initiative. So without further ado, please, Dr. Burman, the floor is yours.
Thanks a lot, Cindy, for inviting me and for hosting this, and please help me to get some comments in so we can be interactive, as you said. And thanks to all calling in. I'm hoping to provoke some thoughts, so please share them with us in the chat. So I will put some slides up asking for comments, but do add comments in the chat all the time. So we will, in this talk, we will kind of take one step back. We have some different topics that's pretty much are about borrowing, some other things as well, and it's touching pretty much around kind of foundations of statistical inference and how we should think and what are the limits for what we can do. So we're starting with why would we need to borrow information from outside a randomized trial or outside the population, subpopulation that we're interested in. And I think we should look a bit at this uncertainty principle. It's not really that we can investigate everything. We cannot get answers all the time. You may have to borrow a bit or guess a bit and trying to do what's best. So we're starting with perhaps the simplest example I can think of. We have two arms. We have an active drug and we're testing this versus placebo. We have N patients. We have a simple endpoint, dead or alive, and we can think of this response as being relatively immediate. We don't have to wait for a long time. We call the mortality P1 for active and P0 for placebo, and we're thinking about a risk reduction of 20%. So the first question to you is, how many patients do we have to plan to die in our phase 3 trial to give us 90% power? So do we need 1 death, 3, 1 per 10, 30, 100, 300, 1,000, 3,000, 10,000, or whatever? So how many patients should we have to power the trial to give 90% power? And we can think about the mortality as being relatively low, and we were talking about the 20% risk reduction. So the first thing I would like to say is that I feel that we often are thinking we are jumping into simulations too early. We're not really trying to see the big picture. So approximating is a very, very good advice. So in this case, we have two treatment arms. We were talking about mortality, dead or alive. That's a binomial distribution. If the probability of dying is pretty low, then this is kind of a Poisson distribution. So if you're looking at the control arm and saying that we have 100 deaths in the control arm, we have a Poisson distribution. The variance is the same as the mean. So the standard error is square root of 100, it's 10. So we have a plus minus 20 as the confidence interval. So if you should compare this to another arm, and we also like to have good power, it's far too less to have 100 deaths in the active group. And we can do this with some simple mathematics. So I'm writing up the estimates and the variance, blah, blah. And then I get an estimate that is 1000 times 1 minus p divided by p. So this means that we need 1000 patients divided by the mortality risk in the trial to get 90% power to detect a 20% risk reduction. That means that we roughly need 1000 deaths in this randomized controlled trial. And it's pretty similar if you're doing a Poisson approximation directly, if you're using a log rank test, whatever p is as far as it's low. It may not be 1000, it may be 850 or something like this, but this is the magnitude. So let's pause here. Who are we to design a trial to try to get 1000 deaths in the trial? That means, say that we have 500 deaths kind of in the control group. We have a 20% risk reduction. It's 400 on active arm. So that means that we have an excess death, 100 people dying on the placebo group because we are not giving the best treatment from the very beginning. Should we afford to have so large clinical trials? And I think this is partly about the ethics within the trial. And to me, the fundamental thing, and I've been working with an ethicist trying to, many years ago, trying to articulate this, is that it's a bit simplistic to say that we have equipoise, that we don't know which treatment is best, because if we're good patients, we always have a hunch. So to me, it's ethical to design the trial, to work with this as a statistician and design this. If I would be okay to let my old mother participate in this trial. If I felt that she would be much better off to be outside the trial and get the treatment that's the best that's available, then I don't think it's okay. I don't think it's ethical to randomize other people into this trial, even if you are talking about informed consent, because it's not very easy for a patient to assess the risks, but we are in a much better position to do that. I guess that you may disagree on this, and then please put your comments in the chat. A comment a bit aside here is, would it help to have a response adaptive randomization? So I've seen a lot of papers about response adaptive randomization, and they have been along for many, many years. It was a lot of this when I was a PhD student more than 30 years ago. And I say that, yeah, that could help in the trial, perhaps. But if you are increasing, if you are randomizing not 1-to-1, but 2-to-1 or 3-to-1, in most cases, this is driving up the variance for a certain number of patients in the trial, which means that it's taking longer to get enough information. And then we are denying patients outside the trial to get the best treatment. So in most cases, response-adapted randomizations are not doing much good. Of course, you could have another than 1-to-1 randomization rate if you need to get more safety information. And sometimes it could be optimal to go away a bit from 1-to-1. So if we're going outside the trial, then we are thinking about the wider ethics. So you could argue that why should the state interfere in this? Why shouldn't you be able to take whichever drugs you want to? And I think this is a bit of, we can have a comparison to have being a very liberal politics about narcotics. It's probably making the society much better that we have some kind of regulations, and we probably need some kind of regulations for drugs, for pharmaceuticals. That said, it's not so, everything is not so black and white. It's not so clear cut, and we will come into what do we really need as proof of evidence. And would that be the same in all situations? So we are statisticians. We know that we are very seldom being 100% certain of these kind of things.
Dr. Burman, there's a comment in the chat box
yes,
asking questions. Are you assuming that both treatments are freely available? Suppose that new treatment is not available and in short supply, and the only chance the patient has to get the treatment is to enter the trial. Equipoise no longer applies.
Yeah, so I think that we should probably restrict new drugs. So we are not allowed in general to get new drugs unless they have been tested in a randomized trial. So I do think that we should have the regulations, we should have these randomized trials, and we should try to get the answer as soon as possible. But in the current situation, we have a lot of cases where this drug is available on the market, so it could be used off-label. So that's complicating some things. So one way of trying to a bit mitigate the problem is that we are trying to close the trial as soon as possible. And I think that we should use group sequential designs more often. And perhaps we are using group sequential designs pretty often when we can. And then I think that perhaps we should be more aggressive than using O'Brien-Fleming. Obviously, this is not just about proving efficacy, it's about the benefit risk assessment. And I don't know, and I was saying that you have some of the answers. So would it be possible to continue the trial to gain more information on efficacy and safety while the regulators are reviewing interim data having a statistical significant efficacy? So if you have some ideas on this, please put it in the chat. So do we have these large randomized clinical trials having 1,000 deaths? So we have had, here at my city, we have had a lot of cardiovascular outcome trials, and then we could very well have more than 10,000, sometimes much more than 10,000 patients. And often we are targeting something like 1,000 events, occasionally 1,000 deaths, but of course, it's much better if it would be okay to use major cardiovascular events than having to wait for patients dying. In oncology, I think we often have 1,000 patients in the trials. There are some that's much larger than this, but I think that 1,000 is a typical value for a large trial, a couple of hundreds otherwise. It's quite common to work with not 20% risk reduction, but rather 30 or a hazard ratio of 0.7. So that's decreasing the numbers needed a bit. And then often we're looking at progressions for ethical reasons. Even if we're looking at overall survival, it's more difficult to motivate a trial where we are really targeting this and don't get too much problems in the current events. But so I think that we should have good standard. We should try to get enough evidence for approving new drugs, but we are in very, very different situations. So in smaller populations, in subpopulations, or for rare diseases, we cannot wait. We cannot have trials with 1,000 major events. So then I was thinking a bit about, shouldn't we be a bit explicit about how much could we ask sponsors, how large trials could we ask from sponsors? And then I was playing around and saying, okay, if we would say that we could, if we're taking the total treatable population, kind of the market that we would have, we and our competitors, and for in some instances we have treatments that with short duration, so perhaps we should look at how many patients, not the prevalence but the incidence, and look over 4 years or whatever. And then I was thinking about, if you're taking the total population and then we're taking the square root of this, would that be a kind of an approximation for how many patients we could have in a phase 3 program? So if we're looking at the COVID vaccine, we had the order of 10 billion people. Is it 8.1 or something like that? The square root of this is 100,000. And I think the main Pfizer vaccine trial had 44,000, and that's not patients, but individuals. If you're looking at other things here, what do you say? Is this the kind of sample size that would be possible? So please give some comments. And of course, what we're talking about here would not be the required sample sizes, but this is kind of the maximum sample sizes that regulators could ask sponsors to feasibly do. And you could look at this from another angle and see what would make sense economically for sponsors. So we wouldn't want to be in situations where we get neglected diseases where it's not worthwhile at all to develop a drug. I kind of felt that the square root perhaps was a bit easier to get for rare diseases than for common. So perhaps we should have another exponent than 0.5. But of course, this also depends on the complexity and the circumstances of the trial. But in whatever we're thinking about this, I guess that we would probably agree that we cannot run equally large trial independent on the size of the patient population and the other factors. So for rare diseases, for smaller subpopulations, there could be limits for what we can do. And perhaps we can have 2 independent trials with a hard endpoint for common diseases, asking for p-values less than 2.5%, but perhaps that's not really feasible for smaller populations. So a possible solution, of course, is to have a wonder drug. And sometimes for rare diseases or ultra-rare diseases, we may actually have a wonder drug. It may be that the disease is due to one genetic defect, and perhaps we can fix this, and perhaps we can see very much larger effects than 20%, and then perhaps the problem is solved. Of course, we're trying to, we could have endpoints that's much more informative. A 0-1 endpoint is never very informative. So could we have something, a continuous variable? Could we use win ratios or surrogates? That would help a lot. It could also be helped a bit by using crossover trial, or helped a lot if we really could use crossover trials. And I think that we are a bit overly conservative and not seeing the full potential and being too concerned about carryover effects that we could probably model away in many cases. We could think about pragmatic trials as well. But at the end, we have situations where I think that we have to relax the requirements for proof of efficacy. So perhaps we should accept type 1 error that's higher than 2.5% in certain situations. And in a way, I think that Bayesian borrowing of historic data will be doing something like this. So as soon as we're borrowing information from outside the trial, and we're kind of getting help by this, by reducing the sample size, that will lead to an inflation of the traditional type 1 error. And this is Kopp-Schneider who has most clearly shown that. Cindy, do we have any comments in the chat now?
No more comment.
Okay, you should wake up a bit. How controversial do I have to be to get some comments? So I would like to give one example where we are thinking about using borrowing. And I will start by sketching this, and time permits, we will come back to this a bit. This is ongoing work together with Darren Scott from AstraZeneca Cambridge. And we are thinking about a situation where we have a clinical trial with 2 subpopulations. So we're thinking about this as biomarker positive and biomarker negative patients, B and C. It could be other situations as well. So we are working in a model where we think that we are from the beginning thinking that we should have, if we don't have any efficacy in B, we will not have any efficacy in C. If you don't have effect in biomarker positives, then this is not a useful drug. But we may have similar situations in other cases. So perhaps we are pretty confident that we should have, depending on the scale that we're measuring, better efficacy in severe disease than in moderate disease. And then severe could be B and moderate could be C. And OA here would be all comers, the union of the disjunctive sets B and C. And I've been asking a few regulators about what they think about the need for showing evidence in the complement population C. And then they, a bit surprisingly, often not start by trying to answer that question, which is a tricky question, but they are often emphasizing that they would really like to see data in this total population, because perhaps we have some efficacy, perhaps the drug is useful also in C. But will we be able to detect that? Will we be able to make that kind of conclusions? So if you're going to multiple testing, there are a lot of things that we can do. We can split alpha, we can utilize the correlation between A and B when we're testing. This is based on the bar. But one common way of testing this is that we run a hypothesis test in the biomarker positives. And then if that is statistically significant, we can recycle alpha and we can test also in all comers at the same total family-wise error rate alpha, 5% two-sided or 2.5% one-sided. But if you're thinking a bit about what do we mean here, what are we really doing? So if this is a placebo-controlled trial, we start by checking, can we prove that the drug has a positive effect in the biomarker positive subpopulation? So is the new drug, is our drug better than placebo? Say that now that we get the statistical significance, so we are kind of saying that we have been proven the drug to be effective in the subpopulation B compared to placebo. What we're doing then in the testing procedure is that we are testing the null hypothesis of no effect in the total population. So we're saying that we know that we have positive effect in B, and then we're thinking about, do we have zero efficacy in B union C? And if that would have been true, then placebo would be much better than our drug in the complement population in biomarker negatives, which I find quite unlikely. Okay, we can have drugs with toxicity giving negative effect, but if you're separating efficacy from safety, then I wouldn't expect the drug to be worse than placebo. So it's kind of stupid what we're doing. So I'm kind of a multiplicity expert. I get a lot of questions about multiplicity, but sometimes it's like, why do regulators ask us to do things that are pretty obvious? Why are we testing in all comers when we know that we have effect in, say, half of the population? On average, of course, we will have effect in A as well. That doesn't mean that we have effect in the union. Of course, it's not saying that we have effect in C. So that is the question. And in many cases, that's not very easy to demonstrate because we're talking about a smaller effect in the subpopulation. So please put some comments in.
Yeah, there are comments from Stefan. Perhaps you can unmute yourself.
Hi, Kaffa, Stephen here. So when you asked a question about borrowing, I mean, there are different types of borrowing. You could be using historical control data to augment the concurrent controls. Or you could, for example, take the point of view that the effect in women should be mildly predictive of the effect in men. So you have some weighted estimator between the two there. That's like a sort of interaction borrowing rather than a main effect borrowing. Or you could even say, well, I have a lot of historical data on covariates. Why don't I try and improve the business of estimating the effect of covariates on the outcome to improve the control in the current trial? So I think there are lots of different scenarios. The riskiest, in my opinion, is using historical data to augment the concurrent control group. That relies on very strong assumptions.
Yeah, I agree. And we will continue with kind of all the examples that you mentioned. So we could, if we are borrowing control group data, then we will have a type 1 error, but perhaps we need to do this sometimes. Perhaps it's worth taking that risk. using female data to say something about efficacy among males. That's a bit similar to this situation, that we have two disjunct subpopulations, B and C. Can we use them to borrow between each other? And of course, we can use covariates, so we can learn about the model from earlier data, and then we don't need to get a type 1 error. And I will give an example from a prognostic score methodology doing exactly this, if we have time.
Okay, just to give a quick example, bioequivalence studies are an example of borrowing from one population to another. Healthy volunteers, but the results will be used. Healthy male volunteers, but the results will be used in elderly, sick females.
Yeah. So we also have the great problem of extrapolation from the randomized trial to the actual decisions about prescribing in the real world. So, Stephen, we're looking at some more slides, and please come back with the same again about this at the end of the presentation. Okay?
Do you want to continue with the comments, or you want to save some?
Yeah, please.
There's another comment from Fredrik. Fredrik, do you want to unmute and talk?
Yeah, hi, Carl. This is Fredrik. This is just a comment on your point earlier
Hi.
on group sequential designs. I mean, if we could do this thing of submitting based on a registrational end, but then continuing following up, I think that would sort of broaden the applicability a lot. I think the challenge there is maybe sometimes are the results post positive interim interpretable? The other idea I think is for overall survival, that is fine. For other type of endpoint, there might be some concern, but generally, I think it helps the group sequential designs a lot if you don't have to meet all the endpoints at the same time. So if you can kind of, so I think yeah, that would help in your example you had earlier there.
Yeah, great. Thanks.
One more comment from Bob Beckman. You want to unmute?
Sure. Yeah. So, hi, Coffee.
Hi.
So, I pretty much agree with what you're saying, and but I wanted to actually broaden it. So, in my comment, I ask, why wouldn't this apply to common diseases? Why do people always assume that it applies only to rare diseases? And my point is this: in many instances, in the real world, patients can't get their drugs because they're too expensive. They're either too expensive for the national health system, or if it's in Europe, or they're too expensive. The insurance won't pay for it, and the people have to pay their rent and buy food. So they forego these medicines, and they suffer morbidity and mortality. And quite a few people are affected by this. And so it seems to me that certain reasonable flexibility of trading off statistical rigor versus public health benefit in the real world, not public health benefit in a math equation, public health benefit in the real world should be considered for some of these innovative approaches in common diseases as well. And health authorities tend to say, economics is not their bailiwick, and they can't do anything about it. And as a result, I think people suffer. So can you comment on that?
Yeah, at least you're inspiring me to share some kind of related thoughts, and I think it's extremely tricky to understand health economics, because we're having so very different systems in different countries. But I think it's important not just to think about two stakeholders, the regulator and companies. I think that we have to have a societal perspective and saying what's really good for the patients at the end. And we have to factor in the cost of the drugs, as well as efficacy, patient-centric views, safety, efficacy, and trying to see what is the best for this individual patient based on her characteristics. So it's a very complex pattern. But if I don't know so much about this, but trying to be perhaps provocative again, would it be possible in the US system to have insurance that are not covering any drug, but covering one drug for this disease? So I think that you are sometimes getting a few drugs on the market, but we don't know which one is best. So we don't run head-to-head comparisons. Wouldn't it be possible to run pragmatic trials in US in cooperation with the insurance companies, say, to find which one of these 10 different drugs on the market is best, and also to use that to factor in the price and the competition?
Yeah, well, that's certainly something that might help. I mean, I think that as you may know, if you follow what I'm doing, I have an ulterior motive, which is confirmatory basket trials and the definition of multiplicity that is applied to them. And so, but I think, there are many potential innovative approaches. And I think to me, the issue is that the health authorities have gotten used to making these judgments for rare diseases, but they don't feel it's their prerogative to do it for common diseases. And I wish there was a way around that.
No, and I understand that there are certain regulations and laws in different countries, but I'm interested in thinking about how would we build a good system of regulations. So I think that we should not always take FDA's views as granted. I think we need research in how should we build the regulations, the laws. But
Yes.
I think we're jumping to, thanks, Bob. Could we jump to David now, Cindy, and then I will try to continue.
Yes.
So, David, please.
Dave.
There's quite a lot of this discussion seems to rather presume that the new drug will be good, but many, especially in oncology, aren't good, and or certainly not better than the preceding ones. They're Me Too drugs. The trouble with when people talk about real-world effects, I'm afraid that too often they mean confounded effects that aren't real.
No, so I think this is related to what I talked about. Couldn't we get more head-to-head comparisons? So first, I think it's unethical to run a cancer trial with placebo as a control if we have a good treatment on the market. So I can compromise a bit with these kind of things. Sometimes you could perhaps think about checking objective responses after 4 weeks and then giving the best treatment. And in many other situations, the patients could very well volunteer to help us generate information. But yes, I think we should understand that there are a lot of drugs that do not have efficacy and perhaps even more are so toxic, so they shouldn't be on the market. Therefore, we need the regulations, we need the phase 3 trials, and I think also that we need to compare Me Too drugs with each other in a better way than we're doing now. So I'm continuing now. Please put more comments in the chat. Perhaps we wouldn't have time to talk about everything, but you can check the chat. and I will try to address some of the questions I've already got in the coming. So we can say something, summarizing this up a bit. We have a logic of reactions, and this is not for the so we're going back to, okay, we're showing an effect in biomarker positives, you're showing effect in all comers, but that doesn't mean that we have an effect in biomarker negatives. And I think that sometimes we are we get the statistical significance in A, and then we get the drug approved in A, and that's not really the obvious logic here. So actually what we are doing is that in this trial population, that we're testing in, when we get a statistical significance, if that's not a chance finding, the implication is that there are some patients in this RCT population which have positive efficacy. And then we kind of need other types of expertise and wisdom to go from some individuals have efficacy. Will everyone benefit from having this drug? Should this be approved in C? How much efficacy would we have if we're trying to extrapolate out to real people? And this is an important question for the payers, especially in the European system. But I think we are thinking too little of this, and we are kind of being too simplistic. A statistical significance in the population does not mean that we have efficacy in all the individuals in that population. So a bit what we perhaps should want regulators to require. So we need some kind of proof of efficacy, and I think that we cannot show efficacy everywhere, but we can have one relevant endpoint. We can have, we would like to have a low p-value, of course, but the requirements here may have to be relaxed if we have challenging situations. We could use Bayesian borrowing to strengthen and supporting this evidence, but I think we need some robustness in this, and we shouldn't trust only mathematics. We need subject matter expertise in doing this. But this proof of efficacy, to me, this is about hypothesis testing, or the Bayesian equivalent. And we should try to enhance the power here. Whereas the next step, looking at benefit risk, we can think more about estimates and estimation, because then we are more into the trade-off between benefit and risks, and looking at different subpopulations, who should the drug be available for. But we can start with this very clear hypothesis testing, and then we're moving over to benefit risk when we no longer can have proof. It's more of estimations. I thought I should say something extra about the max combo test, and to be provocative, this is a really bad test, and I think that it's suffice to say that in one part of this would weight the first step with a 0 weight. So the reason I was taking this up here is that we need to have a deeper understanding for the fundamental inference principles. So it's not about having the mathematics saying that we're controlling some kind of type 1 error. We need to understand what we're doing. And what we're actually doing, this is to show a benefit somewhere. We're not testing whether two treatments are the same. We're testing if the new drug is the same or worse. So the evidence, the opposite to this, the logic is saying that we do have a benefit, and this is the first regulatory requirement. So now if we're looking a bit, and this talk is kind of mainly motivated by this paper where we're looking at Bayesian dynamic borrowing utilizing historical control data to augment the RCT control group. And we're also looking at what we're calling digital twins, or a company has been labeling Procova, or we're sometimes talking about prognostic score methodology. But for the borrowing part here, so what we're trying to do is to take historical controls, we're putting this together with the RCT controls, and then of course we're gaining power, we can reduce the sample size in the RCT. And that's, perhaps we need to do this for pediatrics or rare diseases or certain situations, but there is a problem with this. So we're looking at a very, very simple way of doing this. And this is, yeah, perhaps I should go back a bit, and Stephen was saying it's different kinds of borrowing. So, so we have a situation where we are running single-arm trials, especially in oncology. And this is going back to, we know that in most cancer indications, the objective response rate for placebo would be 0, basically, around 1% in a lot of trials. And if it's not 1%, then clinical expertise should be able to tell us that. So we can have single-arm trials. The Bayesian borrowing of control group data could use power priors or meta-analytic priors. We will focus on putting a prior for the difference between historical controls and RCT controls. And the different methods aren't that different. They have the same kind of issues. And we're taking a very, very simple example here as a bit of simplistic, but I think it's illustrating the issues. So people have been saying that we can use this Bayesian dynamic borrowing, this kind of mathematical black box, to let the data tell us whether we can borrow or not. So in the optimistic scenario, historical controls are the same. They have the same mean as the trial controls. So capital delta here is 0. In the pessimistic scenario, they are kind of unrelated. So if you look at the historical controls, we have no ideas about what the mean would be in the clinical trial. And then as a compromise, we're saying that we are 50%, we have 50% weight for the optimistic scenario and 50% weight for the pessimistic scenario. And then we're hoping that data should tell us the truth. So when we were looking at this model, and we have a bit different scenarios here, but with the blue curve, this is showing how much we're trusting the informative component. Then we can use all the historical data and treat them as if they had been in the trial. In this case, we are thinking about having the same number of controls historically as we're having in the trial. And then we have the blue curve. What we have on the x-axis, this is the observed difference between the controls in the trial and observational controls. This is in standard error units. So we could have a pretty large difference here, 3 standard error units, and still we would be 50%, what should I say, sure about these being the same and being useful. And if we're looking at in this scenario where we're starting with a standard error of 3, so we have a pretty large difference between historical control and trial controls then, and we're simulating what will the weight be. So in how, to what extent will we borrow the historical data into the trial data? And what we see here is something that's pretty close to uniform distribution. So it's completely random to what extent we are using historical controls. And this is telling the story that no, we cannot build a mathematical black box and hope that the data will tell us if the control group data are useful or not. This is a bit strange conditioning here, but in this case, with our conditioning that you could read about in our paper, we see that the type 1 error is going from 2.5% to a maximum of around 9%. Not very good. But we were starting with saying we don't have enough data in some situations, so perhaps we need to relax the type 1 error, perhaps we need borrowing. And I think then this is not just about statistics and mathematics, this is about carefully looking at the different data sets. Should we expect any differences? And I guess in the problem with Bayesian statistics now, many Bayesian statisticians are that they are not Bayesian enough. So if you're going into Bayesian statistics, do go back to the good old days with real subjective Bayesianism and trying to utilize all the knowledge that we have to see, could we take the old data and use them to help in the new trial? And I guess in many cases, that would mean that, yeah, we're thinking that over time, standard of care is getting a bit better. So the responses in the placebo group should probably be expected to be somewhat better in the trial than in the previous trial where we're using data from. Stephen was saying something about utilizing covariates. Then I have an example where we have treatment and control, and we don't see much of a difference. But if we could have a doctor predicting what's happening, and if we're comparing the outcome with this pre-specified covariate, doctor-defined covariate, then all of a sudden we have a separation and we can get the statistical significance. So what people are thinking about, perhaps we could have an AI, machine learning, to learn from old data what the best prognostic score would be, and then we're plugging this into an ANCOVA. And I think this is a great idea. The problem is, do we reveal in dependencies that we couldn't see ourselves? And to me, the solution is probably, yes, we should use old data. We could very well use machine learning, but perhaps at the end of the day, we're helped to see and get some insights in how covariates could help us. So perhaps we don't need to have a random forest estimate. Perhaps we can have something simpler. Going back a bit in the last minutes to this borrowing between subpopulations. We have been working on a mathematical model where we're coupling the responses in these two groups, and we're saying that the ratio of this we're putting a prior on. And the results here is this is the coupling parameter, and then we're comparing with the same color, we're comparing the prior and posterior. So what this is saying is that the data, again, that we get will never, in interesting situations, overwhelm the prior. So we need the external, the subjective prior will determine how we're interpreting the results. So yes, we can borrow, but we shouldn't just borrow in terms of having an algorithm for with Bayesian dynamic borrowing, we should go out to ask experts about this and the interpretation of data in these kind of situations. So say in this biomarker negative subpopulation that you have half of the efficacy in the biomarker positive, it's not significant in its own right, but then it's a discussion about do we expect that does this make pharmacological sense that some of this efficacy would carry over, and then based on that kind of reasoning, looking at the safety, then we can do the best we can to make a recommendation about should patients also in the biomarker negative group receive this drug. And of course, in many cases, it's not really 0-1 negative or positive. Perhaps this is more of where should we put the cutoff. So some kind of summary or conclusions. There are limits to what randomized clinical trials can do. We should try to use all the relevant data, and sometimes we need to borrow information. If we're going down in smaller and smaller subgroups, of course, we cannot get statistical significance in each and every subgroup. So we need to utilize some understanding of the disease, what the drug is doing, to help us make wise decisions.
There's one comment before.
Yeah, sure.
Steve, you want to unmute yourself?
Yeah, I sort of tend to think that it's implicit in the way in which we regard information as growing as we get more numbers in a clinical trial, that we are relying on the fact that bad luck at the beginning could, to a certain extent, be cancelled out by contrary results towards the end. That implies that we are essentially exchangeable about the order in which the news comes. We would regard a trial with 70% success at the end as being the same if it had been all failures at the beginning and all these successes only came at the end, or vice versa. We regard those two things as giving us the same information. If you follow that to a logical conclusion...
This is the sufficiency principle on this slide?
Yeah, that's right, yes. But if you follow that to its logical conclusion, it means you can't really incorporate historical data unless you have the same historic attitude towards historical data. So my view is you can't peek at the data to see whether they agree or not. And then if they agree, and only if they agree, decide you're going to use them. Because it's sort of implicit that you have to be prepared to average the two, otherwise it doesn't work. Yeah. So I'm very suspicious about these sort of alternative algorithmic approaches. I'm not sure they're really Bayesian.
No, I think that we should be more, we should go to experts. We should try to see that, yeah, we have this old registry and saying, who knows if they are very representative for what we're doing in a randomized trial 10 years later. It's pretty useless. But here we have data on a very similar randomized trial that was failing. But now we're doing a new trial with another drug, the same sponsor, the same centers. It's pretty likely that the patients will be pretty similar. So we're borrowing that kind of information. We're putting that into the test, and then we get the statistical significance. But of course, we are a bit concerned about, well, should we really trust this? So then we're looking at the pure data in the randomized clinical trial, and perhaps we don't get the statistical significance at the 2.5% level, but we get a p-value that's 6%. And then when discussing around this and seeing that in this small population, it wouldn't have been very easy to double the size of the trial, perhaps in that case, we would be fine and say that, yeah, it's probably working in this population. And then we're looking at safety, of course. So I think that we need some pragmatism, and we need to build on science, and science is much more than statistics.
There's one comment from Bob again, agree with Stephen's comment, the dynamic.
I'm afraid we're running over time, so thank you to all who've been listening in, and of course you're very welcome to continue if you want to. So,
Yes.
Bob.
Well, perhaps I'll just
yeah. So I just said that I was basically agreeing with Stephen, but putting it in a different way, and then he in turn agreed with me. And basically, I said it is circular logic. In other words, if you're using a power prior, you may be only using the historical data to further confirm what you would have concluded without the historical data. So I was just agreeing with Stephen, and then he agreed, and he sent a link. And then I will also say, someone said the last person you want to consult is an expert, and I said that the picture that you used of me as a physician is first of all, I didn't give you permission to use that picture of me, but seriously, having
So Bob,
having been asked to play the role of an expert, I would say that experts are overrated. I would sort of agree with the other point that was raised.
I think, Bob, that we should have the requirement that the expert is a professor both in oncology and in statistics.
Yeah, you might not have too many experts, but
no, it's you will be the expert for everything.
Well, that's what I'm afraid of, because when I've been consulted in the past, I haven't. My crystal ball has been rather cloudy, but people have been insistent that I say something. So anyway.
All right. This is a fun session. Thank you, everyone. Kathleen, you want to have a last to say, or before we close?
My last words. Thank you very much.
Great. Thank you, everybody, for joining. As I mentioned in the chat, if you would like to receive the invite for the future and you haven't received this one, please send me an email and we'll include you in the future webinar invites. Thank you so much. Thank you, Dr. Burman. Have a great rest of your day.